I'm going to talk to you today about osmosis. In osmosis, we have a solvent that moves through a semi-permeable membrane. What is a semi-permeable membrane? Essentially, it's some kind of a barrier that allows some molecules to pass through but keeps others behind. A really great example of this is your skin. Your skin is a semi-permeable membrane, and some things can pass through your skin and be absorbed by your body, and some things cannot. So when we talk about the solvent in osmosis, most of the time we're talking about water. And so what the water will do is it will move from an area where there's less solute to where there's more solute. So essentially what it's doing is it's equalizing the concentration of the solute. If you look at the two pictures here, you see a concentrated sugar solution on one side of the semi-permeable membrane and a dilute sugar solution on the other side of the semi-permeable membrane. So what will happen is the water molecules here are shown in blue. The sugar molecules are shown in red. The sugar molecules are the solute. And the water molecules are the solvent. The sugar molecules are too big to pass through the membrane, but the water can move. And what will happen is the water will move in order to equalize the concentration. So if you look at the left picture, there's a whole bunch of water molecules on the right, very little water on the left. So the water will pass through, and actually what happens is the water passes through in both directions. But more water molecules pass through to the left than come back to the right. And so you end up with equal concentrations of sugar on both sides as compared to the water. And when that happens, you notice that the volume on the left side of the semi-permeable membrane increases. And what happens then is something called osmotic pressure is created. So osmotic pressure is the pressure that is created by this increase in volume on the left side of the semi-permeable membrane. Osmosis happens naturally through a semi-permeable membrane. You don't need any energy, extra energy to make it happen. Maybe you've heard of something called reverse osmosis. A lot of times this is how water can be purified, through reverse osmosis. And it's a process where you actually have to use some energy in order to get the molecules to move in the direction that you want them to move. So it's actually the opposite of osmosis. In the picture, you notice that there's salt water on one side and fresh water on the other. Now, in osmosis, the fresh water would move into the saltwater side to equalize the concentration. In reverse osmosis, what happens is pressure is applied to the salt water, forcing the fresh water to move out and for the water to move through the semi-permeable membrane, leaving the salt behind. What I'd like you to do now is take a moment to pause the slide, pause the video, and read the two bullet points here. Decide for each of them if you think it's talking about osmosis or reverse osmosis. I hope you've paused the video and restarted again. Let's look at the first one. When you have a solvent that's moving from a 3 molar potassium chloride solution into a 1.5 molar potassium chloride solution, the solvent is moving from where the solution is more concentrated to less. That would be reverse osmosis. When the solvent's moving from a 2 molar potassium hydroxide solution into a 3 molar potassium hydroxide solution, that would be osmosis. As the water moves through, the concentration of this solution will decrease. Osmotic pressure is created when the solvent moves from one side of a semi-permeable membrane into the other side. If there are more solute particles in a solution, then the osmotic pressure will be greater. When we look at, for example, calcium chloride compared to glucose, if we had a calcium chloride solution, the calcium chloride dissolves into 3 separate ions. If we have a solution of glucose, the glucose molecules are held together by hydrogen bonds, but as the water separates those hydrogen bonds, the entire glucose molecule stays intact, and so the solution of glucose has only 1 particle for each glucose that's dissolved in the solution. So if we have a mole of glucose dissolved in the solution, we'll have a mole of glucose molecules. But if we have a mole of calcium chloride dissolved in the solution, we'll actually have 3 moles of calcium chloride particles because they'll each separate into their individual ions. So on this one, 1 mole of the substance is equivalent to 3 moles of particles. Over here with the glucose, 1 mole of glucose is equivalent to 1 mole of particles. What I'd like you to do is pause the video, read the question, answer it. When you're done, restart the video and we'll see how you did. So the question was, which will create the greater osmotic pressure, a 1 molar solution of calcium chloride or a 3 molar solution of glucose? Well, we really need to look at osmolarity in order to decide this. So if we have a 1 molar solution of calcium chloride, as you saw on the last slide, that'll dissolve into 3 separate particles. So that gives us an osmolarity of 3. If we have a 3 molar solution of glucose, it dissolves as 1 entire molecule. So that gives us an osmolarity of also 3. So which will create the greatest osmotic pressure? Neither one. They're equal. Okay, pause the video again and try this problem. When you're done, restart it. Which solution will decrease in volume? You've got 2 solutions separated by a semi-permeable membrane. One solution's 7.5% glucose, and the other solution is 2.5% glucose. And you notice I put in their percentages right here. You could tell that because this one had more particles in it. And we want to know which solution will decrease in volume. In other words, which way is the water going to flow? I always think of water flowing from high to low. Now that's kind of opposite as to what we said before, but from where there's more water to where there's less water. So where is there more water? On the left-hand side. It'll flow to where there's less water on the right-hand side, and that means the volume of the right side will increase, causing the concentrations to equalize, so you end up with a 5% glucose solution on each side. The water flows from high to low water volume, but it's actually moving the opposite way if you think about concentrations. So it's moving to where the concentration is more concentrated. So the water flows to the more concentrated side, but that more concentrated side is where there's a lower proportion of water. Osmosis is really important in our bodies. All of our cells are semi-permeable membranes, and so let's look at the effect of osmosis on blood cells. When we talk about osmosis, we talk about different types of solutions. A hypertonic solution has more solute outside the cell. So hyper means high amount of solute outside the cell. Isotonic has concentrations inside and outside the cell that are the same. And hypotonic means that there's less solute outside the cell. So if you think of hypo meaning low solute outside the cell. And you see those depicted here, where the dots represent the amount of solute. Well, cells are mostly water. So if we put a cell in any of these solutions, which way will the water flow? If we put a cell in a hypotonic solution, there's going to be more water outside the cell than in. The water will flow into the cell to try to equalize it. In an isotonic solution, the exchange rates will be equal, so there won't be any difference in net volume of the cell. In a hypertonic solution, more water flows into the cell than out. Now remember what we said before about how in a semipermeable membrane, the water actually flows both ways, but in a hypertonic solution, you're going to have more water flowing into the cell than out. Now think about what we just talked about as far as volume and osmotic pressure. In a cell, if you have water passing into the cell, that means the cell is going to increase in volume. If water passes out of the cell will decrease in volume. And then they can end up looking like this. You see the blood cells on the left are all shriveled because they were put in a hypertonic solution where the water passed out of the cells. The cells over here on the right are all plump and fat, and in fact they can get so fat that they can burst. That's cells that have been put in a hypotonic solution where the osmolarity of the solution is lower than the osmolarity of the cell. And then in the middle we have the isotonic situation. When those cells shrink and shrivel, we call that crenation. When the cells get big and fat and possibly explode, we call that hemolysis. There are a couple of common IV solutions that are isotonic with blood. One of them is normal saline solution. Normal saline solution is a 0.9% mass per volume solution of sodium chloride. Another is commonly known as D5W, dextrose 5% in water. That's a 5% mass per volume solution of dextrose, or otherwise known as D-glucose. Your blood cells are not 5% glucose, but the reason the 5% glucose solution is isotonic with blood is because it exerts the same amount of osmotic pressure as the solution inside your blood cells. Now, keeping in mind that a 0.9% solution of sodium chloride is isotonic with blood, what do you think will happen if a blood cell is placed in a 2% sodium chloride solution? Take a moment to answer, and then come back to the video. I hope that you thought about it in this way. If your blood cell is normally 0.9% isotonic with sodium chloride solution, and you put that in a 2% sodium chloride solution, that means that the water will flow out of the blood cell into the solution to try to equalize the osmotic pressure, or to try to equalize the concentration of the solutions. That will not equalize osmotic pressure. What that will do is it'll create less pressure inside the cell. The cell will shrivel up, and you'll have crenation happening. In osmosis, we talked about the fact that the solvent is moving to a place where there are more solute particles, or we think of the water as moving from where there's more water to where there's less water to equalize the concentrations. In diffusion, we're talking about the solute particles instead of the solvent. So diffusion is when the solute moves from an area of high concentration to low concentration. Think of putting a drop of food coloring in water. It starts as a little blob of food coloring, and it spreads out throughout the water because the molecules are moving and pushing it around. That's what diffusion is. When we think of a person who has kidney disease and their kidneys can't function properly to clean their blood, oftentimes that person will undergo dialysis, also known as hemodialysis. Hemodialysis uses the properties of diffusion and osmosis together. It pumps the blood out of the body, and it goes through this tubing. It has a dialyzing coil, and it passes through this dialyzing solution. Now, the solution doesn't have a very high concentration of things like salt and waste products such as urea that your kidneys are trying to clean and that get into your blood. So those small molecules, those small solute molecules, can pass out of that dialyzing coil and that dialyzing tube and into the dialyzing solution. In the meantime, larger particles, colloidal particles like albumin and starch, can't pass through the membrane, and so they stay and they're pumped back into your blood, into your body along with your red blood cells. You might know that ions like sodium and potassium and calcium are really important for our bodies. You hear about drinks that are supposed to help keep your electrolytes in balance, and if your child is really sick, you might buy them something like Pedialyte to help get their electrolytes back in balance. How do these ions move across our cell membranes? There are 3 different modes of transportation. The first one is passive diffusion. In passive diffusion, the small particles can simply pass through or diffuse through the cell membrane. The second is facilitated transport. In facilitated transport, the particle needs a channel to pass through. Oftentimes, we're looking at polar molecules moving through a channel that opens and closes. You might recall that the cell membrane has an outside and inside polar layer with a nonpolar layer in the middle. So these polar molecules need a channel in order for them to pass through that nonpolar layer. The third way that particles move through our cell membrane is by something called active transport. In active transport, the molecules move in the opposite direction that you would think that they would move. They move towards an area of higher concentration, similar to reverse osmosis, where you need energy to push the water through to get it to a place where the concentration from a concentrated solution to a less concentrated solution, now we're talking about the solute, but we want it to go from where there is less solute to where there's more solute. So it takes energy to make it do that. You might notice in this picture that facilitated transport is called facilitated diffusion. Those terms can be used interchangeably. If you look at the cell in the little white box on the left, what we want to know is which way will the sodium ions move if we have facilitated diffusion, which is sometimes called facilitated transport. So I'll do that in blue. In facilitated diffusion or facilitated transport, the solute moves from high concentration to low, and the only reason it's facilitated is because it needs a channel to go through. So oftentimes ions like sodium or chloride will need a channel. Things like glucose also need a channel. So in facilitated diffusion, the particles will move into the cell. Now, if we had active transport, that's when our solute moves from an area of low concentration to an area of high concentration. Think of it as if you were trying to shove yourself into a room where there's a whole bunch of people. It's hard to push yourself in there, so you need something to help you. Maybe your friend behind you has to give you a shove. That's what active transport is. In our cell over here, if we want to make this active transport, I'm going to try to do that in red, the molecules or the ions would be moving out because this is the area of low concentration and this is the area of high concentration. Maybe you're wondering why active transport happens. It's easy to understand diffusion because, molecules naturally go from a high concentration to a low concentration. Active transport happens a lot in things like neurons. In neurons, you have membrane proteins that are constantly pumping ions in and out to get the membrane of the neuron ready to transmit electrical impulses. So here's a question for us. A solute is being transported across a cell membrane into a cell. We want to describe the concentration of the solution outside the cell as hypotonic or hypertonic if the solute is being transported by each of the 3 types of modes of travel that we've talked about. So let's take passive diffusion first. In passive diffusion, we would have the example on the right where if the solute is going into the cell, it would have to be coming from an area of higher solute concentration to an area of lower solute concentration. Now, is this solution surrounding the cell hypertonic, hypotonic, or isotonic? Well, since it's a more concentrated solution, it would be a hypertonic solution. Remember, hyper means high. So in passive diffusion, you would have the solute in a hypertonic situation. Well, what about facilitated transport? Same idea, right? Going from high concentration to low concentration, so that would be hypertonic as well. But if we look at our last example of active transport, that would be where this molecule right here is being pumped into the cell. Remember, we're talking about the solute being transported into the cell. So if this is being transported into the cell, that's where active transport is occurring. And of course, we need energy for that to happen. And as that's going into the cell, we're also wondering, well, what's the solution surrounding the cell? The solution surrounding the cell, in this case, is hypotonic because it's a low concentration. Based on concentration gradients, we should be able to learn by osmosis, but it usually takes more work than this. Have a good day.